From the College of Computing Informatics. So over to you, Anne.
Okay, thank you. Well, I'm very happy to be here. I've never given a talk before at BOSC, so I'm a little nervous. I hope you'll bear with me. I'm here today to introduce you to Integrated Genome Browser. It's been around a while, so you may have already seen it in action. But I'm going to try and give you a very quick introduction to it in case you haven't used it before. So, Thomas, you gave such a beautiful, beautiful introduction just to the problem of visualization and how important it is in biology, and I just loved that. And I want to add just a couple more thoughts, which is that when we're building visualization applications, where we're going with that, what we want to do, is we want to stimulate our users to be creative. We want them to enjoy themselves as they're doing their research, and we want to stimulate insight. So that's really the most important thing. That's what we're really going for. And I want to, when we talked about publication a little bit, you know, those of us that write these applications, you know, our work contributes to many publications. The images from our software show up in papers. So it's actually not very difficult for us to really understand the impact of our work, because we can read these papers and we can read about the discoveries that people have made, and we can get a sense of how different aspects of our software are influencing the discovery process. So I just wanted to throw that out there for kind of meta publication, so to speak. And going along with that, we need to support all aspects of the scientific process. So Thomas, you mentioned the static figures. That really hit me, because in the IGB project, we put a lot of effort into making it feasible for people to make these nice static images, outputting different kinds of formats, making it really configurable. But we never really thought about it making those figures interactive before, so that was really great to hear. Also, we need to think about publishing data and making the process of exploring the data really easy and satisfying. So we've got to think about the user experience in building visualization software. So in the IGB project, that's what we try to do. And IGB, Integrated Genome Browser, it's a Java desktop application. So you download it from our website, you install it on your computer, just like you would do Cytoscape or IGV or any of the other important and well-known Java applications. And you download it and you run it locally. So you get to take advantage, or we as developers get to take advantage of the full spectrum of interactions that we can implement using the Java programming language. So it's Java, not JavaScript. And some of the benefits from the user's point of view is that it's relatively simple. Once you've installed it, once you've got it set up, you can just use it like any other desktop application. You can open files, you can save files, you can do all kinds of things. But we also try to make it easy for people who are sharing data, who have big databases and data files and so on, to distribute those data. So we have a system called IGB Quick Load that was invented very early on in the project more than 10 years ago. And it's basically a very simple system of setting up a bunch of files on a website. If you can configure Apache, if you don't even need anything really fancy, you just set up a bunch of files in the right place with some metadata files, XML and plain text, and that's all you do. Now, it can be a little challenging for some people, so it's not something an ordinary user would do. It's something a data provider would do. And so in my lab, we're using this to distribute a new genome assembly for blueberry, Vaccinium corymbosum. I work on plants. And down there at the bottom is an example of what a user sees once you set this up. And very early on, we added the ability to import a favicon, so you can have a little icon representing your institute. But this is just an example of how that works. It's free, of course. You can get it from our website, bioviz.org. And the main mechanism we're using to distribute the software for users is Java Web Start. And the main advantage of this is that whenever we release a new version of the software, users will automatically get the updates. But there are other ways to get the software as well. We're using Git these days. We started out with CVS, and then we moved to Subversion, and now we moved to Git. And we're hosting it on Bitbucket for various reasons. Partly it's because we're kind of following in the footsteps of the Galaxy project, which I'll talk about in a minute. Also, we've started a new thing. We're now doing using Jira and a public issue tracking system. So we've switched over to using agile development methodologies, and we're trying to be really public about that so that people can track what's happening with the project. So whenever I give a talk or present IGB, people always want to know, well, there's lots of genome browsers out there. What makes this one different? What's unique about this one? Well, it's really hard to say that, because I'm not God. I don't know every single genome browser out there. I just kind of know the ones I've heard about, and that a lot of you have heard about. But the thing that I've noticed that's pretty unique about IGB is that it does something really important, which is fast, fluid, animated zooming. It's also highly interactive, and also it has dozens and dozens of features, many different types of features. Also, we've implemented a way for developers to add new features using OSGI framework. And I won't have time to talk about that. I just wanted to mention it because we're very happy to accommodate new developers. So if you have an idea for a plugin you'd like to add, a new feature you'd like to add, we would love to help you. And we have people who can help you. So I mentioned it does fast, fluid, animated zooming. And what that means is you go from the chromosome level down to the base pair level really quickly. So I'm going to try something really ambitious, following Thomas's lead, and show you IGB in action. So when you first start the application, here's what you see. Human genome, these are all shortcuts to different genomes. And of course, the human genome is probably the genome we're all most interested in, so she's front and center. And then in my lab, we mostly work with Arabidopsis. So what I just did is I just loaded the Arabidopsis genome, and I cheated a little bit. These are gene models. They're very, we're looking at an entire chromosome, so they look really tiny. And I loaded them from my local computer. But normally what happens for a user is that they get their gene models from our server, from our main IGB quick load site. So let me show you how zooming works in IGB. You notice there's a gray line? That's called the zoom stripe. And this is something pretty important because a lot of genome browsers, they don't allow you to set the zoom focus. And this is a concept that has been around for a while, but it seems to be needing to be reinvented over and over again. But let me explain. When you zoom, usually what you're trying to do is go from a larger view down to a smaller view. And how many times have you tried to do this in a genome browser and the thing that you're actually trying to look at zooms out of the way, scoots out of the view? That's called losing context, and you don't want to do that when you're zooming. So IGB and applications similar to it implement focusable zooming. So you can't do that in other applications. Whoops. So now I'm down to the base pair level. And notice I can zoom in even further, and notice what happens to my zoom stripe. Now it's transparent, so now it acts as a pointer. So I can move it around and point things out and make pictures. And then of course now I'm at the sequence, now I can also view the sequence. Also, it's important to be able to interact with sequence. So in IGB, you can click on things, you can select them, you can copy them. And that's what I'm doing right now. I can copy the selected genomic sequence to the clipboard, and that's really important. Another aspect of interactivity is you need to be able to explore. So typically what happens in a high throughput kind of experiment is you have a lot of data and you do some kind of you're testing some kind of condition, like for example an RNA-seq experiment. And you'll end up with 100, 200, 300, maybe more than that differentially expressed genes. And as you're sorting through all of those results, you need to be able to very quickly get information about them. So that's what we have implemented here. When you right-click on something, you can do a Google search. You can search Google to find out more about it, or go to a special organism-specific database, like TER in this case. Okay, so now I'm going to go back to the presentation. And we talked about that. Another element of IGB I wanted to mention is that we really put a lot of effort into the interaction part. So for example, you can click on things and count them. I mean, it sounds really simple, doesn't it? Click on things and count them. But that turns out to be really useful when you're looking at, say, reads that seem to support a number of introns and alternative splicing and so on. And so that's one thing that I wanted to mention about IGB that's fairly unique. So there are many more features I don't have time to talk about. And I want to mention that there is a good reason that there are a lot of features. That's because the application has been developed since the early 2000s, and we've had a lot of people contribute over the years. So up at the top left is Greg Helt, who wrote the original version while working at Affymetrix. And the original aim of the software, as it was developed at Affymetrix, was to accommodate big data. So how many people remember tiling arrays? I don't know if anyone even uses them anymore. Yes. Well, the reason this application was developed in the first place was that we needed a tool that was highly interactive and enabled exploration of tiling array data, but could also accommodate the rather large data sets that we were getting from tiling array data. So Greg came up with a lot of innovations that other people have also sort of reinvented, or maybe they kind of copied his ideas, but some of them were random access file formats, being able to load part of the data from a file. And we've continued to build on those ideas over the years. Okay. All right, so now I'm going to switch gears and for my remaining couple of minutes, talk about another way that IGBY is turning out to be really useful these days in the sort of context of big data and genome science. And that is, this is another feature that's actually really old. So when Greg first developed this software many years ago, our idea was that we wanted to link the software to Affymetrix's website. So we wanted people to be able to go to probe set pages, click a button, and then go to IGBY and visualize the alignment of probes and probe set design sequences onto the genome. And so he implemented a REST-style API as part of IGBY. And it was actually really easy to do. All we needed to do was get Java code, that essentially a web server, build it into the IGBY software, and then boom, it was very easy to implement. So IGBY has a REST-style API, and the way this works is when it launches, it starts listening to a port on localhost. And you can send it requests via URLs. It's actually very simple. And so the easiest thing to do is to tell IGBY to go to a particular location and to load a data set. So how I use this in my data analysis process is, like I said, when I'm doing an RNA-seq data analysis or something like that, I'll end up with a big list of genes that I need to look at one by one sometimes. So I'll write out one of these sorts of spreadsheets in R. I use R for data analysis, using a library that lets me embed links in the spreadsheet. And those links are IGBY links. And so I can just send that spreadsheet to my collaborator, and she can open it up and click on the links. And if she's running IGBY, IGBY will go to the right place and show her the scene that I want her to see. So we use this all the time. And it doesn't have to be in spreadsheets. It could be in a web page, for example. And here's the last thing I want to talk about, which is something we did fairly recently, in which we linked IGBY to the Galaxy workflow system. So I love Galaxy. It is incredibly easy to use. It's a beautiful web-based interface to lots and lots of different command line tools. I mean, I use command line tools all the time myself. I mean, I can type really fast, and I know what to do, and so on. But sometimes when I'm trying out a new tool, and I don't really want to invest all the time and effort in getting it to run on our cluster and whatnot, I'll just go over to Galaxy main and see if it's there. And if it is, I'll try it out. I'll test a few different parameters. It's just easy. It's wonderfully easy. So what we decided to do was link IGBY to Galaxy. So the way this works is we worked with the Galaxy team, and we worked with their external viewers API, and that went really well. And the way this works is a user, like me or you, goes to Galaxy, runs some data analysis. And in the process, they build up a bunch of files in what's called the history, their history. And you can make multiple histories and save them and share them with other people. It's really fun. As you make those files, you can click on the name of the file, and a little box will open up, and it'll show you all the different kinds of visualization applications that you can use to look at that file. And so what we've done is we've added a new link that says display in IGBY. You click that link. The next thing that happens is that link gets redirected to a web page on our website, the biovis.org website. And that page is running a little JavaScript program that checks to see if you're running IGBY or not. And if you've already launched IGBY on your desktop. If you're not running it, it displays a message that says, hey, welcome. Would you like to download the software? Would you like to run it? And then you can decide whether or not you want to do that. But if you are running IGBY, then you see this. You see a message that says welcome and lets you know that your data are being set up and sent to IGB. Then you go to IGBY, and then you work as if you had just opened the file on your desktop. That's exactly how it feels. And a new track appears, and you can work with it. And it acts just like any other file that you've loaded. from your desktop or from the web. You can save it as a different format. And this is something that I discovered recently, which is that I've been looking for a GTF to bed converter forever, and I always end up having to write my own, and it's so painful. And I realized that IGBY, because it can save files in bed format, it can load up a GTF file, and then I can save it as a bed format file. So something I found to be kind of exciting the other day when I was playing with this feature. So basically, the idea is that you can use Galaxy to explore all these wonderful bioinformatics tools, and then you can visualize the results in IGB and experiment with different parameter settings if you want. We have a user's guide, and most of the features, or all of the features, are described in detail. We have a gallery of images that we've made and that others have made using the softwares to give you a sense of what kind of visualizations you could create. And then here's our Jira site where we're implementing agile development methodology, which we haven't done before, so this is new for us. And so you can see what we're working on in our current sprint, what our backlog is. You can look at all of our user stories. And notice, this is for Titus if he's in the room. We have an epic called technical debt reduction. Yes. And believe me, as you can imagine, a project that's more than 10 years old has a little bit of technical debt that needs to be reduced. But it's not because we were bad software engineers. It's because many times in the past we had to implement our own solution for a particular problem that then a third party, that another group, 2 years later implemented a solution for, and we were kind of just adopting other people's more robust, better tested solutions for problems we had to solve long ago. So thank you so much, that's it. And I'd be happy to take any questions if there's time.
Great, so. Great, thanks very much, Anne. So do we have time for one question? I can hear the coffee outside, so I don't want to
we can do the questions
So
at
yes,
coffee.
if you'd like to catch up with Anne at coffee would be great. Oh, is there an announcement? No?
Oh, hello. Suppose that we are in the future where everyone is depositing his own data.
Yes.
On this. It's a broad question. It seems to me that it's very nice to have this kind of figures which are very interactive, but who is going to maintain the services behind the images?
Who is going to what?
Maintain the services behind the images. Who is going to pay and maintain the services? Because it's a technical problem.
Oh, yes. I think it will be, I don't think they will last forever. I think all software has a finite lifetime. It has an expiration date. We don't like to think about it, but it's true. So unfortunately, as brilliant as Thomas is, sorry, so I hate to put you on the spot. As brilliant as we all are, we're not going to be around forever, and someone is going to have to maintain all of that. So they are, to a large degree, finite, which is why the print medium is actually really important, because print paper is a lot more stable than digital media. Ask any librarian, they will tell you. So we can't walk away from print just yet.
Okay.
We can debate it over coffee, I guess.